everyone, today we'll be talking about virus cultivation and diagnosis. It is important because it allows us to diagnose viruses to develop the appropriate treatment options, take the proper public health measures, and survey the disease trends and even conduct further research. You can store viruses in a number of ways. And these methods depend on how long you want to store them for. If it is at 4 degrees on wet ice, frozen gel pack, or in the fridge, then you are looking at storage length about 1 day. If you want something more long term, you can try dry ice or a special freezer set at -80. If you want a permanent storage option, you're looking at liquid nitrogen around -200 degrees. For some viral vaccines, you're looking at freeze drying. So you can use the frozen sample and then dehydrate it using a vacuum. It is very important during this process that you avoid physical or chemical inactivation. And these are the enveloped viruses are especially susceptible to physical and chemical inactivation. So you can use a buffer transfer medium to protect it. And it is also important that when you do freeze drying or low temperature storage that you avoid repeated freezing and thawing, because the crystals that form during the process can actually damage some of the viral structure. To gain access to these virus samples, you would have to know what disease syndrome it causes so you know where to take it from. So if you are having a respiratory infection, you're looking at something like a throat wash or nasal secretion. If it is an enteric or stomach infection, you're looking at fecal samples. If it is something like meningitis or encephalitis, you're looking at the cerebrospinal fluid. If it is a vesicular rash, you're looking at vesical fluid. And if it is something like systemic fever, you're probably going to take their blood. In terms of viral detection, you can use direct electron microscopy to visualize its morphology. And this is taken after you roughly purify the contaminated samples. And then you can use antibodies to stain particular structures or features of the infected cell as well. Or you can use something like viral cultivation to grow it, and then you would be able to have more viruses to look at. Alternatively, based on its preferences and which mediums they grow in, you can also infer what type of virus it most likely is. So this is a very rough estimate, and there are a number of mediums that you can use or cell cultures, including primary cells, diploid cells, or transformed cells. The thing you need to know about the different medium is that for primary cells, they are from the animal. For diploid cells, they have a limited lifespan, but for transformed cells, they can continue to be cultured indefinitely, so they are a favorite option for many. And over here we can see a chicken egg, and the different parts of the chicken egg can be used to cultivate different types of viruses. So if I told you that, there was viral growth in the chorioallantoic membrane, then you would infer that this could be HSV, pox, or vaccinia. But if it grew in the amniotic fluid, this could be influenza or mumps. So viral cultivation allows you to gain a vague idea of what you're looking at, but it is not very specific. To be more specific, you can look at the viral protein antigen, or the viral gene, or the viral genes. You can look at the viral genes, either RNA or DNA, using PCR, which is very sensitive and specific, but and it is also very fast, but this process is prone to contamination. You can also look at the viral proteins, but this is a very complex assay. Nonetheless, it is highly sensitive. There is the risk of false positives though. And then you can also look at host antibodies. And these are very specific, but you have poor sensitivity and the risk of false negatives. Something which is more advanced for looking at viral genes is called a viral chip. So this is just you put the viral gene onto a chip containing lots of different sequences, and these sequences are then read by a computer because the viral genes would be complementary to some of these chip locations, and they would be fluorescently lit up, and these read under a computer gives you a specific reading and tells you what type of virus you're looking at. Alternatively, to quantify, you can also quantify the amount of virus you have by using plaque assay, a quantal assay, or hemagglutination assay. So hemagglutination, if it lyses the red blood cell in clumps, then you have like a shield formation, and that indicates the presence of a virus which destroys red blood cells. If there is a red dot, then that means there is no virus, or there is an antibody bound to the virus. Okay, so thank you so much for listening, and see you next time.